And here we are. And we have our exam, third exam a week from today. And so we're dealing with our last chapter. We should be able to finish it up, or very close to finishing up what I want to talk about for the third exam. And so remember, try to bring questions to class. We can deal with them today or next Tuesday. And we have a assignment due today, and it's back to the nucleus with that assignment. And the assignment is just a video. Our book covers the assignment material in chapter 6. You do not need to look at chapter 6 for any reason. Just it's all contained in that video. But just to be aware that our book covers that topic as well. It's good for us to cover it in the nucleus because that's where it occurs, and RNA interference occurs just outside of the nucleus. And so we have our exam, and each exam is worth more credit like that. And remember, we're emphasizing that soon after our third exam, we have our final. So it's really worth it to work on this material and understand it. And I think it's also good to do the path that we've done in our course to get to this cell biology, because it's very visual, very physical, very organized. Nucleus, protein synthesis, pathway trafficking, cell membrane, all related topics and interconnected topics, yet also topics we can move physically our mind from one place to another. And so just we've covered that, and yep, and that, and so this is where we're at. So we're covering chapter 14 by going through 5 stories, and we started talking about the third one a little bit last time. Okay. So I just want to show you that for that first story, the book does, bring up aquaporin as a transmembrane protein of water channel, and we emphasized it as an example, to put it in example, to really emphasize that trafficking of proteins and trafficking of membrane that happens. And we did the same thing with neurotransmitters. Our book covers neurotransmitters some in our textbook, and we told a similar story in class. And so you can see the neurotransmitter containing vesicles docking on the cell membrane in the presynaptic neuron, and they're carrying that neurotransmitter that we're getting released, and then there's going to be that recycling. And so now on to receptor-mediated endocytosis, and in this context we'll be thinking about heart disease, and that means we're going to be thinking about blood vessels and our circulatory system. And what it's going to be about is it's about LDL, and LDL carries cholesterol through our blood, and that's because cholesterol is very hydrophobic. So you don't need to know the structure of cholesterol, but you need to know is, that's a really hydrophobic molecule. It has only one hydrophilic group on the whole molecule. So it's got to get carried around. And here's a good way to understand the story of molecules like cholesterol, okay? So cholesterol is really hydrophobic. This is just, salad oil and water in a jar, and you can shake it up, and those phases will separate. And one phase is the water phase, and one phase is the oil phase. And people call the water phase an aqueous phase. They call the oil phase a hydrophobic phase. And we can add cholesterol to that and shake it up and see where the cholesterol goes. Where do we find the cholesterol? And the answer is it's almost all in the oil phase. And so the term scientists use is the cholesterol partitions into the oil phase. And in this just states that there's actually a tiny, less than 1% of cholesterol could be found in the water phase. And that's just because they're in dynamic equilibrium, and the equilibrium lies greatly toward the oil phase. But every once in a while, a little cholesterol dances into the water phase, like that. Here's another example of that, just to see this, right? Estrogen, we talked about estrogen in our course. Estrogen is almost as hydrophobic as cholesterol. Estrogen gets made from cholesterol. And one place we have to deliver cholesterol is to the cells in our gonads, our testes or ovaries, that make estrogen or testosterone. And that's because we can't make estrogen or testosterone from scratch. We have to start with cholesterol, and it's a pretty simple modification of cholesterol to make them. And so there's a destination we can think of when we think of the LDL receptor mediated endocytosis. We can say, oh, one place it's happening quite a bit is in our cells that need to make estrogen or testosterone. Yep. And it actually happens in every one of our cells. Every one of our cells needs some cholesterol, in general, just a small amount. And so LDL is the way to carry it around. LDL is a suitcase carrying cholesterol. I just want to point out for the estrogen, since we're on the topic, you can also put estrogen in this, or testosterone like that, and you shake it out, and 90% of the estrogen will be in the hydrophobic phase. It partitions also with really high selectivity to the oil phase, but not quite as much because, see, it's not got another hydroxyl group, and so that makes it a little less hydrophobic than cholesterol. But estrogen also is not, just floating around on its own. That's why we have this situation where the estrogen receptor is actually inside the cell, because estrogen, testosterone, and other steroid hormones can actually diffuse right through that very hydrophobic cell membrane. So just to bring that up, we got to carry the cholesterol around in this. And this is the structure of the LDL receptor. It's got 3 parts, and we just need to know those 3 parts. It's got cholesterol in the center. So then what are the outer parts of the LDL particle? The outer parts are phospholipids to create a sphere that has a hydrophilic surface and a hydrophobic core. So see those very familiar phospholipid heads and tails? So hydrophobic tails, hydrophilic heads. So it's now water soluble. It's soluble in the blood. And then the last component of 3 that we need to know about is this protein. And we can just call it LDL protein. It has a specific name, apolipoprotein B, I guess. I thought it was A, B. And, but the key thing for that is this is going to provide that selectivity, that tag that the LDL receptor is going to bind to and say, oh, yep, this is what I want. I need to bring in some LDL. So we could just draw the LDL receptor on here, and it would look like this, where here's the LDL receptor, and it's bound to the protein part of the LDL. And it's on the cell membrane. We'll make it a little bit bigger. So it's going to specifically bind to the protein. So really nice structure for LDL. 3 different cholesterol. It has some other molecules in it, but 3 major molecules. Yes?
Sorry, what was the first major molecule?
Cholesterol itself. So the red core is lots and lots of cholesterol. LDL carries around some other lipids as well, but here we go, introducing it. 3 parts. It's got to carry cholesterol. It's got to coat that cholesterol with hydrophilic, surface. So let's use phospholipids, and then it's got to be identifiable. So let's have a protein in it. Okay. So that's the structure of LDL, and it's just going around in our blood like that. And it's what's referred to as bad cholesterol, and we can talk about that. Okay. And so it's bad cholesterol because sometimes it just crash lands on the inner wall of our arteries and veins. And when it crash lands, it literally smears out and makes like a greasy spot. And it's really hard to clean up that greasy mess because we smeared out a very hydrophobic molecule, and it's hard to pick it up. There are some drugs that doctors use now that are almost like detergents to use to try to lift up that greasy stain off of your pants. Instead, it's on the inner surface of your blood vessels. So that's a reason why LDL is bad, is because it could get the cholesterol material can get deposited by accident and stuck on the inner surface of our arteries and veins, and then that plaque builds up over the years. That's not our focus. So here's a picture. It's actually from an intro bio textbook of the LDL receptor in action, and we've already talked through it some. The players are that LDL, the LDL receptor, and this clathrin. And the clathrin is going to help create the coated pit and the coated vesicles. And so we talked about that some last time. And so another thing where we just have a list of players that are involved in picking it up. If a cell wants more cholesterol, needs more cholesterol, it puts more LDL receptor on its cell membrane and it pulls them in this way. And so this is a good example of membrane traffic. We would know, oh yeah, the LDL receptor, when it's newly synthesized, it must have a signal sequence. It must get synthesized initially in the rough ER because it's a transmembrane protein. It's got to get to the surface. Then that's how it gets, gets made, it gets to the surface, and then it's recycled. So there's a recycling process where when it binds LDL, it gets pulled into the cell by receptor-mediated endocytosis, then it gets recycled at the cell surface over and over again, as do the phospholipids in the membrane. The phospholipids of the membrane that get pulled in also get recycled to the surface, like that. And our book shows this, picture 2, and it shows this recycling process of it going back and forth. So it's pulling the LDL receptors that have bound LDL, it's pulling them in by a type of endocytosis, receptor-mediated endocytosis. The pit forms because the clathrin is a protein trimer, and it is both clustering bound LDL receptors and also starting to form that dent. It's just got a natural curve to the clathrin trimer, and it creates that coated pit, and more gets loaded in, more gets clustered and gets loaded in until it pinches off like that. And then it comes in, the clathrin gets uncoated and goes back to play that role again. The membrane fuses with others to deliver the LDL. It gets processed into the cell, and then the membrane goes back to the cell surface. So it's a great example of this protein traffic, membrane traffic going out like that, okay? There are, yes?
Can you explain the difference between the ligand binding domain and then the protein? Are those two
Yeah,
domains?
so that's a good, I often say, oh, that's a good question. Then questions are always great. So the way to think about a domain is to think about our own body. So we have an arm domain, we have a leg domain, head domain. It just means a region that has a specific function. And so when we talk about the LDL receptor, we would talk about 3 functional domains. There's the extracellular domain, and that's the domain that's going to bind that LDL protein. And that's the ligand. That's what we got to bind and hold on to. And then we have the transmembrane domain of the LDL receptor that crosses the membrane. Then we have that intracellular domain. And that's got to bind to clathrin. And so I described before that the structure of the LDL receptor is sort of like this, where my arms are the ligand binding domain, my waist is the transmembrane domain, and my legs are the intracellular clathrin binding domain. And when it's not bound, my body's in this position, and then when LDL gets bound, I go like this, and like a pliers, my body conformation of the protein actually shifts, and it exposes the intracellular clathrin binding domains like that. So clathrin binds to each of my feet, and my feet are together, can't bind to them, then it opens up. So domain is just a generic term for a region, a functional region of a protein that we could name like that, yeah? Yeah, and so, it's got this sweet 3-dimensional conformation that changes shape when LDL binds. And that leads to the binding in the inside of the cell of clathrin to my feet. And the clathrin trimer can bind to one of my feet and one of Priscilla's feet, and, one of Chase's feet, and start clustering them together like that. And then another clathrin can bind to another one of their feet and another one of you, and we get those clustering together. And the feet are only exposed when the LDL receptor is bound to LDL. And so that's kind of the sweet, real world physical story of how can we get the LDL to be clustered. And remember, we're comparing receptor-mediated endocytosis with that other kind of endocytosis that's called pinocytosis, where the cell just engulfs some liquid and pulls it in. And we're like, yeah, that's not selective enough for this task. It also is not going to be able to pull in enough of LDL. So we got to have a specific pull-in mechanism. And there are plenty of other molecules that get pulled in by their own receptor-mediated endocytosis story, like that. A key thing to mention here is, just a side note, is there are several proteins involved in this pathway. We're just learning 3. The LDL protein, the LDL receptor, and clathrin. But there must be uncoding proteins and so forth, like that. Lots and lots of proteins. And any protein has to get made from a gene. And any gene could be contain a mutation, right? In our room right now, we have multiple alleles of the LDL receptor, just like everything else, right? When I was a kid, growing up in my little hometown in Minnesota, in 11th grade, Lenny McCarger's family owned a grocery store, and Lenny McCarger's dad died of a massive heart attack. And he was a young guy with kids in school and working at the store, and he just keeled over. And almost certainly, Lenny's dad had something called hypercholesterolemia. High cholesterol for a genetic reason. And there's a good chance that Lenny's dad had an allele of just any one of the proteins that are needed in this pathway, so that he was not able to pull LDL in very efficiently. And when you hear of someone who has bad heart attacks in their 40s, there's a really good chance that they just have, and we have different alleles, right? You could have it worse or not worse. And there are lots of people who have just genetically high cholesterol. And there are, a variety of reasons why, but we're looking at a pathway that could be the story in some people, like that, yeah? And those people could try to avoid, cholesterol in their diet, but our body makes cholesterol. We gotta have cholesterol even if we don't eat it. So, lots of have medical relevance, right, for this kind of pathway, because it happens in humans, and it would happen in every vertebrate, like that, this LDL receptor pathway, like that. You gotta move around some LDL, because you gotta move around some cholesterol, like that, okay? So our book shows that. I have a couple images that are showing the same thing. Hopefully they just show them, a little simpler, a little more visual, like that, right? So the receptor, it's really trying to show those domains, right? The domains that Spencer just asked about. So that extracellular ligand binding domain, and that would be LDL. And this, I think, is just, I don't know why they have that carrier protein. Maybe there is a linker protein, I don't know. But basically, the protein is going to now bind to clathrin. Clathrin's going to get clustered into a coated pit. And it's going to get pulled in, it's going to get uncoated, and that cholesterol is going to continue to be brought in, and the clathrin is going to get recycled back to the cell membrane. And so, we need to be able to draw and label that for our exam, like with those, relevant proteins labeled, okay? Here's just another image of the same thing, like that. Something to mention is, well, where is this cholesterol going? And the answer is the smoothie R. Right? Because that's where we're going to synthesize estrogen, right? That's where metabolism, both degradation and synthesis of lots of lipids takes place. It's not heading to the rough ER, it's heading to the smooth ER. And how's it going to get there? It's going to get carried in a membrane-bound vesicle. Yep. And so let's just talk about that. Because we can fit this in really nicely to a little bit bigger drawing. And it goes like this. So LDL, those particles get synthesized in a variety of cells in our body, but mostly they get synthesized in the liver. So we're just going to think the liver, okay? And we're going to talk about LDL. That's our focus, LDL receptor mediated endocytosis. But we're going to just put in a little bigger context, because you'll be like, oh, okay, here we go. And And we can bring HDLs in. HDLs are called good cholesterol. So we can just bring those in briefly. Our big focus continues to be LDL receptor-mediated endocytosis. But let's just start out, maybe go over here and talk about synthesis of LDL in the liver. So this is going to be a liver cell. And this is where most LDL gets synthesized. And so in the liver, where are we going to synthesize the cholesterol? So cholesterol is going to be synthesized or processed here, and it's going to be in the smooth ER in the liver. We know that for sure. Okay? Now, where is and again, we're just going to call it LDL protein. Where does that LDL protein get made? And the answer is the LDL protein gets made in the rough ER over here. Okay? And so that's where newly synthesized LDL protein is. And what's it look like? It looks like this, where the extracellular ligand binding domain is in the lumen side, because that's going to eventually get out here, fuse, and put LDL receptor on the surface like this. No, it's not going to put it on the surface of this cell. Sorry. What? Hold on. I started going after the LDL receptor. That's not happening in this cell. This is the LDL protein, and the LDL protein is going to be made in the rough ER, and this is where we're going to assemble LDL particles. So we're going to make the LDL protein here, and we're going to assemble LDL in the rough ER. And to do that, what's going to happen is we're going to get some vesicles pinching off and pinching off with cholesterol and bringing it to the rough ER. So there's going to be cholesterol in that membrane. They're going to join it here, and we're going to build those in the rough ER, and then we're going to secrete them through this pathway. So that LDL protein, it must have a signal sequence on it. It gets synthesized in the rough ER, and that's where we're going to put this little package together. And again, cholesterol getting synthesized, getting processed here. It's got to get over here. It's going to do that in the membrane of that vesicle, get over there, fuse with it. And then we're going to use phospholipids in the rough ER and cholesterol, now in the rough ER, and the LDL protein to put that package together and then release it from the cell by exocytosis. So on the way out, we would have these assembled particles, and we could draw vesicles like this. So there's a vesicle, and in it has completed forms of LDL. It's got that LDL protein, it's got the LDL, it's got the cholesterol, it's got the phospholipids, and it's going to release it by exocytosis into the blood, and it's going to float around like that. So we're making LDL in the liver, and and so that's that, and so that it's going to float around. Then it's going to go out to target cells, like in the gonad is our example, but really any cell is going to want to pick it up. So over here, we're going to have the LDL receptor, and we're going to say call this a target cell. And we're going to say, well, now where did that LDL receptor come from? And the answer is, well, the target cells all have to make it using their gene and synthesizing it, and synthesizing it in the rough ER. And that's going to get synthesized in the rough ER, and then it's going to make its way to the cell membrane by this process through the Golgi and through exocytosis to be on the cell membrane. And then it's going to go and be there and go through that recycling process of binding LDL and bringing it in and so forth. And when it brings in LDL, the LDL is going to be brought in, and that vesicle is going to head to the smooth ER. So it's kind of nice to see the production and the use of it. Production in the liver, and see where, okay, it's got to be in the smooth ER, got to get dealt with there, and then head to the rough ER to get put into an LDL package, leave the cell, float around the blood, get picked up by a target cell, like a cell in the ovaries or testes or any other cell, and then head to the smooth ER to get dealt with there. What happens in the smooth ER in the case of our focus is going to use enzymes to convert the estrogen or testosterone, no, the cholesterol to estrogen or testosterone, like that. Then release that into the blood. Yeah, so it's nice to see that. So then here's just the last thing, is that when there's excess LDL in the blood, or cells can have too much cholesterol, and they can make this HDL particle, which is called good cholesterol, and that cholesterol is heading back to the liver. So we can have excess LDL or HDL, and that's supposed to go back to the liver, where the liver picks that up and sends it to the smooth ER. Sometimes it gets broken down and used to make other molecules and things like that. So just a little, zoom out and see a little bit of that pathway, right? Because in the liver, the pathway in the liver cell makes sense to us. Got to, get this cholesterol going in the smooth ER, got to get it over to the rough ER, got to get the LDL protein and assemble the LDL particle, got to get it to the cell membrane, exocytosis, release into the blood, like that. Our focus is especially the target cell and that receptor-mediated endocytosis. Yep, so just a little bit of that, because again, it's hugely significant in our health. And so I just wanted to talk about that a little bit, and just kind of roll through it quickly, but to give you a good feeling for appreciating, like you could also draw on the exam, the liver cell, and where's cholesterol get made, where's the LDL particle get assembled, where does the LDL protein get made, smooth ER, rough ER. And so, we really are distinguishing between the smooth ER and the rough ER. Yep. Anybody have any questions about that or anything at all? Yeah?
So where is estrogen made?
So estrogen is going to get synthesized in the smooth ER because it's a fat, it's a lipid, and it's a lipid, and it's very hydrophobic, and so it's going to get synthesized in the membrane of the smooth ER. Cholesterol is always surrounded by some hydrophobic molecule with a hydrophilic surface because it's so insoluble, like that. And then estrogen gets made, especially in ovaries and in some other cells. Yeah. All right, so we're going to go to, that's receptor-mediated endocytosis. Got some heart disease, got some relevance for folks in my hometown and every town in America. And, sometimes people have too much cholesterol in their body because they have too much in their diet. Sometimes they have too much and they really couldn't do anything about it, really, because it's the issue with the alleles they have in the many proteins on this pathway of LDL and cholesterol movement, like that. Yep. All right. So now we're going to talk about, we have two more examples. We're going to talk about lipid rafts. And so lipid rafts are bound in the cell membrane. Our book talks about them in chapter 14. And to get to lipid rafts, we just have to talk a little intro bio about cell membranes. And so here's the phospholipid bilayer. All life on Earth is made up of cells. Every cell on Earth has a cell membrane. Every cell membrane on Earth is made up of phospholipid bilayer. So this, structure, universal feature of life on Earth. And we've been talking about it, like some about it, but just to bring that up again, phospholipids. And let's say it was in 1972 where a scientist came up with this called the fluid mosaic model for the cell membrane. And mosaic because that phospholipid membrane is decorated with all kinds of proteins. So this chapter is both about the phospholipids and other lipids and fats in the membrane and all those proteins like that. So that's what we're doing too, right? We're talking about protein traffic, we're talking about membrane traffic. And so just a drawing of that, and it's just a term here, glycoprotein. So that's a protein that had sugar polysaccharides added to it in the Golgi, and it's on the outside. The glycoprotein has that sugar on the outside. Here it's showing cholesterol. Cholesterol would be embedded in that hydrophobic region where the hydrophobic tails are, because it's just so hydrophobic. It'd be partitioned there. You could find a tiny, tiny amount of cholesterol out of the membrane, but it's not sitting there for any length of real time like that. Okay? So, and we've learned about various phospholipids and proteins that fit into that. All right. So here's a picture from our book showing that, and showing examples of that, including proteins like this would be RAS, and this could be any of the cell surface receptors we talked about, the PDGF receptor, and so forth. And here's just to underscore that is the glycoprotein, so a protein on the cell membrane where the glycosylated form is outside of the cell. The glycosylation enzymes are in the lumen of the Golgi, floating around in the lumen of the Golgi, and that's going to become the outer surface of the cell. The membrane, those phospholipids are very fluid. And so these proteins and phospholipids are fusing around side to side really fast. And we've emphasized that about the phospholipids, but these proteins in general are also sliding side to side, like boats that are, don't have a rope tied to them, and they're just floating around in the ocean, and they're just floating around and bobbing around really fast, side to side like that, and diffusing back and forth, side to side, a lot. And so a big part of that is the fluidity of the membrane. So these phospholipids are held together by non-covalent interactions, and they just, those are very weak, non-specific interactions. So there's lots and lots of diffusion. And so for us, they're really just a couple of examples of where cells change the fluidity of their membrane. And they want to do that to hold things together sometimes, like that, or let things move around. And so this is just an example of that, where the fatty acid tails could be saturated. Saturated means they're saturated with hydrogen atoms. All the carbons have single bonds with hydrogen. And an unsaturated fatty acid is a fatty acid that has got fewer hydrogens because it has some double bonds. And so it's got carbon-carbon double bond, and that would cause a couple of the hydrogens to be gone, right, like that. And that's going to change the fluidity of the membrane. And we have a good example of that where margarine is made from vegetable oil, and vegetable oil has some unsaturated fatty acids, and it's more fluid because this structure is not as easy to pack together tightly. And to make margarine, they just hydrogenate, add hydrogen to those unsaturated fatty acids in that, not in phospholipids, but in the oil of that, and it just lowers the melting temperature. And our cell membranes are very much like that. The diffusion of the molecules in the membrane is temperature dependent, and the fluidity is temperature dependent. And one of the factors that regulates that is the types of phospholipids and how many hydrogens are in their tails. Okay? Here's another way fluidity is adjusted in the membrane, and that is the presence of cholesterol. So this is just showing cholesterol, and cholesterol changes the fluidity of the membrane, changes the packing of those molecules, of phospholipid molecules. And the fluidity of the membrane, dependent on cholesterol, is temperature dependent. And so it gets a little bit complicated, where at low temperatures, cholesterol increases the fluidity. At higher temperature, it decreases it. For us, we have stable temperatures in our body, right? Constant body temperature. At that temperature, cholesterol in general decreases the fluidity. So when we say, we have to have some of these molecules and we got to kind of hold them together, one way to do that is increase the cholesterol in that region. And that's what's called a lipid raft. So here, let's just jump to a picture of a lipid raft from our book. So here's an example of the idea of a lipid raft. Is these boats are bobbing around, these molecules are bobbing around and sliding around really fast, and you want to hold these close together. And one way of building a lipid raft is to increase cholesterol in this region. Another way is to decrease the unsaturated fatty acids in that region. And the third way is to include another kind of lipid that we're going to learn the name of. And so lipid rafts have higher concentrations of cholesterol, saturated fatty acids, and this molecule. And it's shown here, and we'll talk about the names of them. It's shown here in our book. And again, I wanted to show this one more time to say you just do not have to memorize the structures or names of these. We're going to have a very short list of these lipids, and it includes it includes cholesterol, and then not for this exam so much, but especially the final exam, but we could bring up on this exam the PIP3, phosphatidylinositol, right, and then the phosphatidylserine. So those are names you have to learn, and cholesterol and like that. Just to show you, again, we're not focused on structure, but these are called sphingolipids, and they're just a different structural biochemical category of lipids that have a hydrophilic region and a hydrophobic region. And this is the basic structure, and that's why they're called sphingolipids. And that's the structure of a typical phospholipid. Each of them will have more atoms here to make that hydrophilic head, but that's the core structure, just so you can see it. And here's another view of it. So sphingolipids, instead of having that two-tailed hydrophobic tails like a phospholipid, they start off with just having one. But then certain of them can get a second one added. So there's the sphingolipid basic structure, and then we have specific examples where they'll have a hydrophilic head and they'll have adjustable hydrophobic tails. So they're not phospholipids, but they look like phospholipids, and they're in the membrane like that, and there are a whole bunch of different kinds of them. And for us, I'd say we're learning about these membrane lipids, right? So phosphatidylserine, PIP3, cholesterol, and then these sphingolipids. And all we're learning about sphingolipids is they decrease membrane mobility. They're found in lipid rafts at a much higher concentration. So when we see a picture of lipid rafts, we say that's a region that simply has more cholesterol, that decreases the mobility, more saturated fatty acids, decreases the mobility, more sphingolipids, decreasing the mobility. And that's a lipid raft, and the purpose of them is to cluster together, group together molecules that should be nearby each other. Lipid raft. So that's what we're dealing with here, and I'll just scroll back to this, is that we're talking about lipid rafts. So our fourth example from the chapter that the book talks about is lipid rafts, and we just want to put them in a context of some examples. And the first one is to talk about their structure. This is just a figure from the next chapter, and I think after this one. No, I think it's in this chapter, and I might just be an old picture. But what's just shown here, just to give you what's the purpose of this figure, it just says, look, we could take cells, like a human cell and a mouse cell, and we could actually join them together. And in doing so, we could watch how the human molecules in the membrane just mingle into the mouse region and vice versa. And we can look at how quickly that happens, and it happens really quickly. So again, that mobility is true. And what we see is those lipid rafts don't move around as fast, right? They'd be the last thing to get, mixed up between the merged cell membranes. And so just some, not from your book, but just to put it in some context, to talk a little bit about lipid rafts like that. And you can see that, there are examples of lipid rafts in lots of different health and disease stories. So this is just going to put them in some context where it's about the organization and the function of a lipid raft. And there would be lots of lipid rafts in every cell membrane, and they would have different lipid rafts for different functions like that. So they're like islands in a rapidly changing sea. And they just help organize activity in the raft. And so here's the kind of stuff that you don't have to scramble to write down, but just an indication of, wow, they're involved in a lot of different things with regard to our health and disease. So lipid rafts are the big focus for research in some really important human diseases. Lipid rafts are often the place where viruses target to get inside a cell. Because of the way they're organized, they just are a good target for a virus. And then, back to our topic of our course, cancer biology. Cancer cells often have disrupted lipid raft functions like that, okay? So just to talk a little bit about this, and again, with somehow the lighting or the projector in here, I just want to highlight, when I have a big paragraph that says, highlight those sentences. So they're involved in protein clustering, energy metabolism, oxidative stress responses, inflammation, autophagy, apoptosis. They're just a really common story. And they would be part of the cell membrane and the function of the cell membrane. And it would just be the clustering of some function in there. It says protein clustering. I should mention that the LDL receptor mediated endocytosis, that's not a lipid raft story. We covered that separately. Yeah. So just another little phrase out of this paragraph. Essential roles in various cellular processes such as signal transduction, membrane trafficking, and cell adhesion. It says here, yes, receptor-mediated endocytosis is often involved in lipid rafts, but our LDL receptor-mediated endocytosis is not one of those examples like that, okay? Then just a little bit about this. This is actually out of order. Sorry, I got that out of order. And so this is just a figure to kind of provide you some images of the idea that lipid rafts play a lot of roles. We don't have a specific example like heart disease where we're going to dive into and say, look at this function of a lipid raft in this example. Rather, just a lipid raft. What's a lipid raft? It's a place of decreased membrane motility because of the composition of the cell membrane. And the composition is based on the lipids in the cell membrane. And in a lipid raft, we have increased cholesterol, increased saturated fatty phospholipids, and increase in sphingolipids like that. So that's going to play the role of creating that lipid raft of decreased mobility. It's a little bit like a little piece of, the last piece of margarine melting in a pan, right? If you put margarine in a pan and heat it up, it's going to melt and it's going to turn into a more liquid oil, but the margarine is hanging in there. That's what a lipid raft is like. It's very weak. It's not a thing that's like, you could get on it and float to Hawaii. It's very weak. It's just held together by a little bit more forces than the rest of the cell membrane. The rest of the cell membrane is so fluid that it makes it really difficult to perform certain functions. And so here's just an image of that. Again, not one you have to worry about getting tested on, but just to show you that in our apoptosis story, that was one that listed, and lipid rafts are a way for those external signals to go into cells to communicate about, encouraging cell death. And that's where part of the story comes for cancer biology like that. Just another images of viruses getting into a cell using a lipid raft and so forth. And that's that. Another slide that's a little bit out of place. So lipid raft is an example that our book covers. I want to cover in here just to bring up its relevance for all kinds of different states of human health and disease, but not a big example. So good examples of all these lipid rafts, short example. Then our last one we want to cover are cell adhesion molecules. And cell adhesion molecules are proteins, and they're in the cell membrane. And those are covered in the next chapter in our book, and we don't have to look at that chapter. That chapter is mostly about other topics, and it includes cell adhesion molecules there. And we're going to just pick a specific example again to roll through our cell adhesion molecules. And so here we go with the final example that fits into chapter 14 about cell membrane, but actually is found in a kind of an odd location in the next chapter in our book. So the first thing to mention about them is just to sort of clarify this confusion about the lipid rafts and the receptor-mediated endocytosis. So for LDL traffic and receptor-mediated endocytosis with the LDL story, lipid rafts play a role. They play a kind of a really interesting role. And what they do is they hold on to extra LDL receptors that can't go into a coated pit. And so we can draw that here, and we can just show it by adding it to this figure, is that there is, we talked about, if a cell needs more cholesterol, it could add more LDL receptor to its surface. However, in some cells, the cell will build a lipid raft and it will hold some LDL receptors there, like a marina, where you dock your boat and it just can't float away. And so it provides a nice way of giving us an example of a lipid raft. Again, we don't have a big example to dive into and say, look at this function of this lipid raft, but our example will just be to connect the LDL receptor story with the lipid raft story by saying, we just learned about receptor-mediated endocytosis as our third example, and in our fourth example, we can add it as sort of an extra feature, and that is that, these LDL receptors are just floating around freely, rapidly sliding side to side like a boat that doesn't have an anchor, and as soon as the LDL binds it, clathrate will bind it, and it'll start getting clustered into a coated pit. But when it's in a lipid raft, it's just not able to float around as freely like that. So even if it binds LDL, it's not going to enter a coated pit. Even if it binds LDL and clathrate binds to it, it's hanging out in this lipid raft. The boats can't move around. The LDL receptors can't move around as freely, and so they can't get clustered and start forming a coated pit. Like that, and that's that, just to talk about that. So, in some examples of the LDL receptor mediated endocytosis, lipid rafts are important. In this case, it is because it's involved in preventing it or reducing it. So that's a nice visual example of a lipid raft and a function of a lipid raft for us. Yep. All right. And so, I'm going to skip that here because that gets more complicated. Lipid rafts, and that was the last piece of lipid rafts. So now onto these cell adhesion molecules, which are all proteins on all our cell membranes. So you don't have to struggle with this. I just want to show you this overview. They're involved in lots and lots of processes in our cells, cell adhesion molecules. And it says up top here, it says cell adhesion molecules are essential surface glycoproteins. So they're proteins on the cell membrane. They're glycosylated proteins. They've got an extracellular domain, transmembrane proteins, intracellular domain. Cell adhesion molecules are made on the rough ER and get out to the cell membrane through exocytosis. Okay? And so you can see there's a list like that, and we're going to focus on one example. Our book talks about it some in chapter 15, but we're just going to deal with it in class. And just again, glycoprotein, got to know that concept and term. And our example is this. So here's some immune system cells that are going to fight infection. So you've got a cut on your finger and it's turning pink and warm and painful, and your immune system is called to take care of it. And there are a variety of white blood cells, immune system cells, that are going to help. And we're just going to focus on one that's called a macrophage. And so that cell is in the blood, floating around, moving around in the blood. And the cell we're going to focus on is an immune system cell called a macrophage. And macrophages would be getting made in the bone marrow every day of our lives, and they, out there, and they're just floating around, and they do a variety of things. And one thing is, here's a macrophage, and it's just floating around with the red blood cells and the other immune system cells, and then it has to leave the blood to go into the tissue. So that's what we're going to focus on, is the cell adhesion molecules involved in this macrophage leaving the blood and crawling into the tissue. Eventually, what the macrophage does is it phagocytizes the bacteria. So it's a really classic fighter at this site. If you have an owie, and it's oozing lots and lots of immune system cells. Where did they come from? The answer is they were in the blood, circulating the blood, and they detected trouble and left the blood. They had to leave the blood. And so the steps are going to be, I think maybe have this. The steps are going to be able to include really 3 of these stories in one. So cell adhesion molecules play lots of roles, and we're going to focus on, in that macrophage story, 3 cell adhesion molecules and the role that each of them plays. Okay? So the 3 are the tissue structure. The cells, the macrophages, have to squeeze between cells to get out of the blood and into the tissue. Then they have to crawl along the blood vessel and find the place to leave, and that's part of an immune response. Okay? And so in those, we're going to learn about 3 cell adhesion molecules. There are lots of them, but we're just going to have one example that we're going to focus on, ask questions about it on the exam, people to describe and draw pictures. And so these are the 3 classes. There are lots and lots of different members of these cell adhesion molecule proteins. So they're proteins, they're glycoproteins on the cell membrane, great ones to talk about in chapter 14. Some reason our authors, moved it to another chapter. So selectins, CAMs, and cadherins, and that's the order they play in the roles of having the macrophages leave the blood. Okay? So again, we're going to look at this, and they're going to leave the blood 1, 2, 3, like that. Okay? So the next thing we have to say is just a blood vessel is lined by cells, and they're called endothelial cells. And our macrophage has to squeeze between the endothelial cells. And the endothelial cells, they make a really tight connection between the endothelial cells, so that it's a tight, they have to keep the liquid in the blood and not having it squeezing out, dripping out, leaking out into the tissue, like that. So we're gonna see those are just like bricks, you know, with tight connections between them, and the macrophage needs to go between them. It's not gonna hurt those cells, it's just gonna slide between them, but doing so, it's gotta squeeze, you know, like between two bricks in a sidewalk. It's gotta find a way to squeeze between that. So that's what's happening. And so here's the flow of it. So the macrophages are floating along like this, and they're like, okay, I'm near trouble. I'm getting the detection, I'm near trouble, so they bind. And their first binding to the surface of the blood vessel is to cell adhesion molecules on endothelial cells. So here are the endothelial cells. This is the blood where the macrophages are circulating. This is the tissue. So the first step is there's trouble, I need to land, like that. And they land by binding to the selectins. Then they're near trouble, so they have to move. And they move kind of by crawling, kind of by rolling. And they move toward the right exact spot by using connections to cams on the endothelial cell. And then finally, they need to squeeze between the endothelial cells when they've come to the exact right location. And out here, these are cells, and they're squeezing between those cells to go hunt bacteria that are in the tissue outside of the blood, like that, yep. So 1, 2, 3, bind, we'll say roll, and squeeze, like that. These cell adhesion molecules play lots of different roles, so many different roles in our body. We're just focusing on one to get a good example of cell membrane function, and our focus is actually about the endothelial cells and what's happening with them. And they have all the time on their surface right now, they have selectins, cams, and cadherins. And the purpose for the macrophage is I can bind, can land on a selectin, I can roll on cams, and I can squeeze between cadherins. So it's a really nice example to show us some features of cell adhesion molecules. Like that, okay, all right. And so our book in chapter 14 does bring up the idea of connections between cells. And this is kind of what we're looking at. And so this would be, in our case, not the intestine, this would be the blood. These would be endothelial cells, and this would be the tissue where the bacterial infection is. And these two cells are connected, are holding close to keep a tight seal so the blood stays in the blood vessel and doesn't leak out into the tissue. And so we've got to bind and roll, and then we're going to squeeze through there. And so that's an image from our chapter 14 of what's going to be happening. The endothelial cells are really held together by two different types of connections, and ours is called an adherens connection, like that. And so just to go back here and show you, this is where our last part is, the cadherens, those are holding hands together to make that blood-tight seal, and this is called an adherens junction. So we should learn that term, and this for us would be, these would be two endothelial cells lining a blood vessel in our body. And they're making a tight seal. And the tight seal is called an adherens junction. And they're holding together because these proteins are holding hands with each other to make that tight seal. So these cadherins are present in the endothelial cells, so these would be proteins in the cell membrane, and they have an extracellular domain, and it's like a hand, and they just want to hold hands with other cadherins on other cells, like that. So that's what we got to squeeze between. And these are super stable. They're stable for our whole lives. These adherence junctions should be stable for our whole lives. And somehow the macrophage has to squeeze between them. So this is the last step in the process, the squeeze between process. And so what we would see is that this is where the macrophage is going to be, and it's going to squeeze. Get very narrow and squeeze between that. And the way it gets through those is that the macrophage is going to be here, and it's going to release enzymes that are proteases. Because those cadherent proteins are holding hands, and they don't want to let go. They're going to plan to do that for our whole life. And so the macrophage releases proteases, and remember we've talked about those. We've had some different examples of proteases, and these are proteases that clip those cadherent proteins, clip the hands off of them. So there's a little gap, and they squeeze through by breaking that hand holding by chewing up the hands, like that. And they squeeze through. After they've squeezed through, the endothelial cells, they're always making cadherents, and they make fresh cadherents to replace the lost ones, like that. And so that's a visual from that next chapter. It's not in this chapter, but just to give you a good visual of that step in the process. And so that would be happening here, right? Bind to selections, roll along cams, and then chew up cadherents and squeeze between. And so we're getting from this is that the focus, although it seems to be on the macrophage, the focus is on these endothelial cells, the cells that line the blood vessels, the inner surface of the blood vessels. And they're expressing these cell adhesion molecules in their membrane that are selectins and cams and cadherins. And in the case of the selectins and cams, the macrophage has cell surface proteins that are binding directly to them, briefly, temporarily, and using them to find their way along that way. And lots of cells do that. We have lots of other cells that migrate, that crawl from one place to another in our bodies, especially during our embryonic and fetal period. There's lots of this cell migration. So in this example, we have some good cell migration, right? It's crawling along, holding hands like that, temporarily, holding on, letting go, and finding the right spot, and then it's squeezing between. Another really good example, and that example is those cadherents. And other cell adhesion molecules are really good at holding tissues together, right? Our heart holds together, our brain holds together, and so forth. So in just one example, we get a good look of this another role of the cell membrane, like that. And so in, again, the chapter we're not looking at, the book talks in a little bit more detail about these. But we have a good story like that. Like that, like that. So that's our good story. That's our last story. That's, I have some of the slides from the chapter, because people will probably have questions about other pieces of the chapter. But this is how I like to, I guess I have to scroll back a ways to find that list. This is how I like to think about how to organize this chapter. And it's good. Again, I'm a molecular biologist, so I don't really care for putting it in context, but really classic context, right? Like that. And I like to say, wow, like our bodies are magical, right? Like that, but so are squirrel bodies, they're just as magical. You can get really excited about, urine production and so forth, but squirrels are doing that too, right? So slow down, slow down for squirrel. But, these are just great, because they bring up features of the cell membrane of how dynamic it is in this pinching off and adding and the membrane trapping, the protein trapping. And so that's where we're at. And like I said, I have some more slides. We can dive into some of those. I have new material that we can talk about next Tuesday if you don't have questions. So right now, do people have questions about anything at all? And so we're doing great. We still have a few more minutes of class, so let's just say that, yes?
I have a quick question. So the selectins, the cell adhesion molecules, and the cadherins, are those all on the epithelial cells, or are any on the macrophage?
There are lots on the macrophage that are complementary. Yep. Yeah. And just to mention, so you said selectins, cell adhesion molecules, and cadherins. There is a kind of a sad, frustrating naming feature of cell adhesion molecules. So cell adhesion molecules are referred to just a big, huge group of proteins. They're not related. They're just all cell membrane proteins, and they're involved in making connections with other molecules, like that. So they're called cell adhesion molecules, and it turns out that then within that, there are some groups, and the groups are gene families, like that. So there's a big gene family of selectins. So we're calling them selectins, but they all have different names like that. Then there's a big gene family of cadherins. And there are some other big gene families. And there's a big gene family, well, it's a big gene family, that are referred to as CAMs. And I try carefully just to call them CAMs because they're actually an abbreviation for cell adhesion molecules. So it's one of these naming things that's confusing, right? Where, selectins, CAMs, and cadherins, they're all cell adhesion molecules. Those CAMs are also called cell adhesion molecules. So it's just, and it's, I don't even know, but it's obviously some historical kind of, reason how it popped up that way. Yeah. But yes, there are lots of cell adhesion molecules on every cell, and there would be cell adhesion molecules on those macrophages already for the selectins and CAMs to do that binding. They're always ready to go. And just to add a little bit to that is when cells are moving along another surface, so they're going to bind and they're going to roll or crawl, that's called cell migration. And cells are doing that because there's some chemical signal, and so the process is called chemotaxis. So these macrophages are detecting a different chemical signal that tells them, land here, you're close to where you need to get to, and then we're going to move to the more precise location, and then we're going to squeeze through. And that squeeze through is just, it's not that the macrophage is using its cell adhesion molecules, it's actually cutting the endothelial cell cadherins temporarily in order to squeeze through that adherence junction space. Yep. Yeah, other questions people have about anything at all? And this is we've talked some about this chapter, right? The book goes through, here are a lot of different lipids, a lot of different cell membrane components like that. There are other structures in there like the glycocalyx that it wants to talk about, and caveoli, right? And so, it's good to ask questions in class. These chapters have spots where the book talks a lot about something, and I'm like, I didn't talk about it, I'm trying to organize it into building a framework like that. But, there could be a couple of questions on the exam that are in the book that we just didn't talk about in class. So one thing you can do that's a good strategy is to just have your book and just say, we need to know anything from page 255. Yeah, and I'm sure I mentioned before, when I had that opportunity in college, I just ask questions. You know, they say, anybody have any questions? I'd say, yeah. What do we need to know on page 255? And they, you know, they explain it. And, oh, well, what about 256? Yeah, and my classmates would boo. You know, I'd sit in front or back, and they would just boo. You know, like, come on. And I'd be like, well, you know, trying to do well in this class, you know. So, you know, you're super welcome to do it, right? You paid for that opportunity, right? One time I calculated like the cost of each class meeting, right? You know, take the class meeting number divided by the, you know, tuition, and that's your cover charge, right? So this is supposed to be a super entertaining time, like you're going to a concert, you know? And so, you know, you want to get your money's worth. And so that's Tuesday. I don't really, you know, I gotta review this, but we should, and fit it in, and, you know, maybe some new stuff will pop up. But otherwise, you know, I'll roll into some stuff that I want to talk about on that, you know, after the third exam, you know, go into some new material that wouldn't be on the exam. Yeah, so that's where we're at. And we'll stop here a couple minutes early, because it's a good stopping point, and we'll just roll forward for our test on Thursday. And, you know, you can see, again, describe it or draw and label it, right? We have lots and lots of examples on this. Cell biology is very physical, very visual, you know. Where does RNA processing occur? And what are 3 steps in RNA processing? Stuff like that. Lots of questions on the third example like that. Okay?